mitochondria are always losing their DNA, then how can they still have their own DNA after all this time?
Well, there were scientists that studied it. They wanted to know how they could prove that mitochondria are losing and retaining certain genes. Here's what they did.
We need to know how to prove that mitochondria are losing or retaining certain genes so that evolution and progressive events can be better understood in today's eukaryotic genomes. It has been a topic of recent discussion in our science communities that there is some sort of evolutionary tactic to the retention and disposal of mitochondrial genes. There hasn't been many strides to prove or disprove that many of the hypotheses that we had come about. Our purpose was to try to disprove or prove, within a reasonable doubt, a few of the more common ones. We chose these two hypotheses: the inferred pattern of mitochondrial gene loss through evolution and the content necessary for predicting mitochondrial gene retention.
I'm hypothesis 1. I stated that the inferred pattern of mitochondrial gene loss through evolution is lost in phases of evolution. I suggest that functions that are centrally important to the organelles have preference. This allows for me to be tested by looking at certain complexes and their functions. The scientists found that the genes that encode the strongest binding energy proteins are the genes that mitochondria have retained most commonly, and this is true across the entire eukaryotic tree of life. This is also shown in the opposite, as genes that are less needed for the central activity are encoded by the nucleus of the cell. You can consider me proven. I'm hypothesis 2. I suggested that both GC content and protein hydrophobicity, which is the aversion to water, are necessary to predict mitochondrial gene loss. I think that finding a link between gene properties, their propensities to be lost from the mitochondrial DNA. Scientists used a linear model to graph the ability of features of mitochondrial DNA to predict the inferred patterns of gene loss. After those results were analyzed, it seems that GC content and hydrophobicity were present in almost 98% of all model structures. The absence of other figures shows the lack of support for other answers. Scientists are adamant that these are completely relative and may not be the most accurate portrayal of what is important to the retentions of genes or which are lost and what they contain specifically. I am neither disproven or proven.
How did they prove this?
Good question.
Well, we developed an algorithm that can identify the ordering of evolutionary events so that we can suggest their causes. This algorithm is called hypertraps, and it's very complicated. To develop hypertraps, we use stochastic modeling with Bayesian inference. Now let's understand what these words mean. Stochastic modeling just means that when estimating the probability of something happening, one or more of the inputs has random variation possibilities. This random variation is usually based on observed or recorded information. In our particular experiment, we examined over 2,000 mitochondrial DNA sequences, identifying the presence or absence of any genes. We then compiled all this data together to recognize the trends that became the basis for the random variation possibilities. Now, Bayesian inference is a statistical test that reports the probability of a certain hypothesis. The advantage of this test is that it keeps updating as more information is added. This means for our experiment, we use Bayesian inference to test the probability of each hypothesis as we discovered more information from the DNA sequences about how and when the genes were lost.
That makes sense, but what about hypertraps? What is hypertraps itself?
Well, we wanted to create a general model for mitochondrial DNA evolution from DNA sequencing. From the sequencing, we were able to infer possible evolutionary paths of the DNA. Following the paths, we noticed gene loss patterns. Then, using these patterns, we created an algorithm, or machine. Then used this machine to find the probability of gene transitions in the DNA. This effectively allowed us to test the likelihood of our hypothesis.
Okay, but how did this let you test the hypothesis?
Because after our algorithm was made, all we had to do was use it and analyze what it gives us. You've already talked to hypothesis 1, so you know that the genes that encode the strongest binding energy are the genes that mitochondria have retained most commonly, and that the genes with lower binding energy are often lost. By plugging this information into our algorithm and testing the result, it allowed us to demonstrate that genes central to the assembly of ETC, or electron transport chain protein complexes, stay within the DNA.
And that means...
That means that there is a set of core genes retained by the mitochondria, and thus it must have a pool of units ready to build a new core gene. Now for hypothesis 2. As you know, our results seemed promising, possibly linking GC content and protein hydrophobicity to gene loss. The problem here is we couldn't find anyone else that has results like us.
And what does that mean?
It means that these results are likely relative. That's why hypothesis 2 isn't exactly true, making it neither proven nor disproven.
All this is great, but what does any of this stuff mean overall?
Even though there has been many theories on why genes in the mitochondria are retained, before now, none of these had been mathematically tested or quantitatively explored. By creating our functional hypertraps algorithm, we created a general method of creating algorithms, or maps, of evolutionary tracks. This is important because it identifies the features that shape us in our modern day. We're also hoping that others follow our example of using mathematics and even our methods to explore and define different hypotheses.